Hi, how are you guys? Welcome to another lecture of GL4 for high schools. Today we're going to talk about studying microbes, especially model organisms in space, and why do we do that at NASA. My name is Sergio Santamaria. I am a principal investigator and research scientist at NASA Ames Research Center in California, specifically in the Space Biosciences Division. I'm currently working on a couple of spaceflight missions, as well as some ground studies using model organisms. I hope you enjoyed this lecture, and please feel free to contact me. My email is down there, and I believe I'll be talking to you during your training. Welcome. So let's start by discussing what are biological organisms or biology going to be exposed once we go to space. And this doesn't affect just humans, as you can see on the video here, one of our astronauts, as well as many other organisms like plants or bacteria or yeast or insects, layer of everything. The two main things that you will hear are, of course, reduced gravity or altered gravity and space radiation. And space radiation is known to be a very dangerous a thing that happens in space. As you can see from the images on the right, you see the Apollo 11 particle tracks from films that were located in different locations in the astronauts on Apollo 11. Every one of those lines is a particle that is crossing to that piece of film. On the bottom right, you can see human cells that are being damaged by different types of radiation. And this type of radiation can be perhaps low effects like gamma or X-rays, but also have very, very heavy effects, especially those heavy particles like iron or magnesium or oxygen. What types of space radiation we're going to find? So there are different types. These are just two of the main ones included here. The main one that you will hear are, of course, the solar particle events or those solar flares or coronal mass ejections that we keep hearing once in a while because we're currently in what is called solar maximum. You also hear of cosmic rays or galactic cosmic rays or GCRs, and these are coming from the universe. They're primarily from exploding supernovas, and there's no way to prevent them because they're always going to be there. Now, we are very fortunate that we have what is called a magnetic field or a magnetosphere that protect us from many of these type of particles. They kind of bounce back or they break apart when they come into the magnetic field. That's why we don't see a lot of damage, perhaps even in the International Space Station. These types of radiation, both solar events and cosmic rays, are modulated by our solar cycle. It's an 11-year cycle. We are currently coming out of what is called solar maximum, which is a period of maximum intensity, and then we'll go towards a solar minimum. During solar maximum, the primary events are solar, what is called primarily protons and helium. And when we're in solar minimum is primarily galactic cosmic rays, or there is an increase in galactic cosmic rays because that magnetic field from the sun is a little bit smaller. They're both unpredictable. However, galactic cosmic rays are known to be significantly more damaging, as I just showed you on the previous slide in the example with iron. So what do we know in terms of biology in space, especially deep space? We don't know much. We do know that we had astronauts going in the 60s and 70s to the moon or around the moon for a period of almost 2 weeks. And we also have data from astronauts that have been uh, in lower orbit, or which is the International Space Station, for over a year. However, we do need these so-called model organisms to give us more information, like guinea pigs or canary on the mine, just to understand what's happening in deep space before we can safely send astronauts to Mars or to live on the moon for a long period of time. Here at NASA Ames, we have done a variety of experiments using automated systems like satellites. There are a few here that all started with GeneSat-1 2006, carrying bacteria, then PharmaSat carrying yeast, then Orios that carry Bacillus, which is a very strong type of bacteria, then SporeSat carry ferns, then ECAMSat in 2017, which was our last lower orbit meaning only a couple hundred kilometers from the ground, from us. And this was carrying pathogenic bacteria to study the effects of antibiotics on bacterial resistance. And then Biosentinel, which I'll talk a little bit later since I'm happen to be the principal investigator. So why do we use model microbial organisms? You might have heard that there are a lot of types of model organisms. There are plants, there are mice, there are primates, there are also bacteria, and there are also yeast. And why do we use them? They're very useful, and I'm going to talk about one in particular since it's the one that I have been working for many, many years, which is Baring yeast. The scientific name of Baring yeast is Saccharomyces cerevisiae. Cerevisiae, you see it on the left. They're small organisms that are approximately 5 to 6 micrometers in diameter. And on the right, you see how they look on an agar plate. So why Baring yeast? Well, it is a eukaryote. What does that mean? We are eukaryotes, humans. What that means is that there are many pathways that we share, especially those involved in the repair of damage or stress response, which are of my interest because that's what I study space radiation. They're very easy to genetically and physically manipulate. What that means is you can go into their genes, you can play with their genes, you can add, remove genes, you can even put human genes in yeast. And of course, assay availability. What that means is like there are a lot of different assays and procedures and approaches that can be used in yeast. Moreover, yeast are used a lot in the San Francisco Bay Area where we are, in the biopharmaceutical, in the biotechnology industry to produce things like ethanol, like fuels, like even plastics, to biomanufacture some things. And I'll tell you a little bit more. I'll tell you a little more later. On the right side, you see examples of my personal examples, like you can use them for biophysics. Those are on the right called DNA curtains. Those little purple things are actually proteins, motor proteins. On the bottom, you can see some examples of immunohistochemistry that you can study DNA repair. And in the middle, I put a little robot that is specialized to use with hundreds and hundreds and hundreds of thousands of yeast types at the same time. What else? Why do we use Baring yeast? There's flight heritage. That's very important for us here at NASA. What that means is I told you about PharmaSat. You see the little satellite up there that carry Baring yeast. And we have done also a lot of experiments on the International Space Station for many, many years and even before, since the 60s probably, if not earlier, using this type of yeast. We can also dry it. If you go to the supermarket, you can buy dry yeast and you use that to make your bread or to make your muffins or whatnot. So you can actually dry this yeast that allows us to have some experiments in space. So even though Baring yeast is a very simple, small model organism, it's one of the best models to do these long-term missions or missions to go to deep space. And I'll show you an example that I'm very familiar with. Something else is very important is how do you go from something so simple? On the left side, you're going to see what happens. These are yeast cells that are in a special medium or liquid growth medium that contains a dye. You're starting to see that the dye changes color. On the left side, those little wells were not exposed to radiation, ionizing radiation to be specific. And on the right side, still kind of remains blue because they have much higher radiation. What that implies is that there is more damage, meaning those cells are much slower at changing the color of that blue dye. This is what we actually do as part of the Biosentinel mission, that you see some of the examples on the right side. So what is important to perform these experiments? You need this instrument, this hardware, these devices that have a lot of heritage in order to have some success in these missions. And a lot of what happens is a lot of the processes involve what do you choose to go to space. In this case, we decided Baring yeast. We selected a variety of strains. How do we fly? Do we fly dry or wet? It needed to be dry. Then in order to do these missions, you also need to know, are they compatible with the plastics? Are they compatible with the reagents? Can they survive in dry state? Then how you sterilize, meaning all these plastics, all these fluidics need to be sterile. Then how many samples you want to use, how many replicates? Do you have controls both in lower orbit and on the ground? And how do you want to send that data back? Many of these satellites are completely autonomous or automated. You do not get your samples back. Biosentinel happens to be still in deep space. Something else that is also very, very important is everything that you can do here on the ground. It does not mean that you can do it in space. You can try, but a lot of the things that matter is mass, volume, energy. So you cannot just use what you have here on the ground. It will be awesome if you can, but many times you have to minimize or miniaturize that type of hardware. So you cannot send your big refrigerator or your big incubator. Everything needs to be miniaturized. And of course, cost is different. The amount of time that it takes to go from the ground to space takes a long time. And I'll be happy to discuss this with you guys another time. So please, and I believe this slide will be available to you as well in case you have questions about this slide. BioSentinel. On the left side is our mission to the International Space Station that launched in December 2021 and returned back to us in August of the next year. And on the right side, you will be familiar with Artemis 1 launch that occurred November 16, 2022, and our payload was one of them. What are the main objectives of this BioSentinel mission? There are two primary objectives. The first one was to develop the capability or the tool to be able to support life or biology in deep space. The second objective is to study the space radiation environment and the effect of this space radiation on biology, on these budding yeast cells. On the bottom, you see the different structures within that satellite, starting with what is called the biosensor payload, where the biology is located. And next to it, we have that so-called LET spectrometer, that is a radiation detector. And everything else around it is what allows this biosensor to maintain yeast or biology in deep space, including, for example, propulsion or solar arrays, batteries, a radio transponder, controls, navigation, etc., etc. The main objective of Artemis 1 was to test the Orion spacecraft. In this case, happens to be unmanned, meaning that there were no humans in the spacecraft, but the goal was to test that everything was tested that went around the moon and returned back to the Earth undamaged. And then during the preparation for this mission many years ago, NASA noticed that there was enough space, volume, and mass available underneath Orion. On the top right, you see what is called the Orion Stage Adapter. This is big yellow ring inside, and you can notice those little boxes. Each one of those is those small satellites, including BioSentinel. Again, this is Artemis 1, and on the bottom left, you're going to see the deployment, the separation of Orion. And after this deployment, a few hours later, each one of those payloads or satellites were deployed into space. BioSentinel was released at approximately 3 hours and 40 minutes after launch and continues to be in what is called heliocentric orbit, which is an orbit around the sun, currently at 40 million miles or 61 million kilometers approximately. And we communicate with the spacecraft once a week. What's inside this biosensor? We do have, I keep telling you, these budding yeast cells. So we put them inside these fluidic cars or fluidic cassettes that are made of plastic or polycarbonate. We dry them inside, we seal those cars, and then we assemble a bunch of electronics around them. And all these electronics are, their main objective is to measure the biology and how they're doing over the course of the mission. Then we assemble the biosensor, and you see in the middle on the left, the so-called LET spectrometer. The LET spectrometer is a radiation detector that allows us to do two main things. One is to detect the amount of radiation, of space radiation that is hitting the box, and as well to characterize the different types of particles, not just from small protons, but also to heavier particles over the course of that spectrum. And on the right, you see the different components of the spacecraft. This biosensor was sent to ISS again in 2021 and returned back to us a few months later. We also perform a variety of tests before we launch the payload, including vibration testing, because we want to make sure that the box is going to survive that big launch. That was a humongous rocket. We also do what is called TVPM, or thermal vacuum power management, which means turning on everything inside the box under vacuum, a very, very cold environment to replicate the space, just to see everything is working properly. Fit checks, we need to make sure that everything fits where it's supposed to be, including the box inside the dispenser from Artemis 1. Now, this is an example of the type of fluidic components. There are a lot of different valves and bubble traps and desiccation chambers, everything so we can fill these fluidic cars I mentioned earlier. We also need to ensure that we can warm up those cars to the temperature that we need for the yeast to grow, which in this case was approximately ambient temperature at around 23 Celsius degrees. Each one of those fluidic cars contains 16 microwells, and inside one of those microwells, as you can see on the left side, we put the yeast. The yeast are located inside the well, and they cannot escape the well because we'll use a special filter so they don't escape the well. And then we measure their growth and metabolic activity using 3 LED lights. We use these lights to measure two things. We use 570 and 630 nanometer lights that allow us to measure the different colors of alamar blue. 570 nanometers measures the amount of pink, and 630 nanometers measures the amount of blue. So we can see the differences from blue to pink, including all those purples in the middle. And then we also have another light close to the infrared at around 850 nanometers that is invisible to the dye, so it doesn't see blue or pink, only measures growth, only measures the turbidity or amount of number of cells that are growing inside the well. There is an example of what we can read. This is after exposure to gamma radiation, which is low power, low energy radiation. From left to right, each one of those curves involves low radiation to high radiation. So you can clearly see how the cells are affected, how much slower they get over time. Now, this is a more representative or space-like environment type of radiation. So we took our cells to Brookhaven National Laboratory in Long Island, New York, where we have what is called the NASA Space Radiation Lab, or NSRL. There we can simulate galactic cosmic radiation. So this radiation is much, much lower doses than what I just showed you before with the gamma rays. It allows us to expose the yeast to something similar, that we might encounter outside the magnetic field, and we see a significant effect even with yeast cells. Here's an example of what we saw on the International Space Station. It's just to show you that each one of those squares is a fluidic well, and you can see the cells growing, except for well number 5 in blue on the left there, because that one contains no biology. But all the other ones contain different types of yeast, and you see how the different LED lights, which are in green, red, and gray, you see the different changes because of biological activity. I mentioned as well that we have captured a variety of solar events over the course of the last 2+ years. Since we launched in November 16, 2022 on Artemis 1, our little spacecraft, our CubeSat, has detected more than 30 different solar particle events, all of those shown in those red arrows there. And this is particularly important because we have passed through what is called solar maximum again. Something else that we have been able to do on the bottom there, you see what is called a spectrum that measures the number of particles and the type of particles depending on this so-called LET. So LET stands for linear energy transfer. It's also known as stopping power. All you need to know is just the amount of energy that a particle deposits on something, in this case on the detector. So you see from left to right, it goes from low energy or low LET all the way to high energy, high LET. Low energy, we're talking about those protons, then helium, then oxygen, carbon, all the way up to titanium and iron. Those titanium and iron particles are more relevant because they're very highly energetic and they will produce more damage on biology than protons. So what have we learned from BioSentinel? Now we're going to talk about a different mission, a different spaceflight mission, but this one is going to the moon. It's called LEIA, and LEIA stands for Lunar Explorer Instrument for Space Biology Applications. Also uses varying yeast and also uses a system similar to BioSentinel. LEIA has 4 primary objectives. Number 1 is to determine the effect of the lunar surface environment on biology. We're going to be using a variety of yeast strains that have mutations on different DNA repair pathways, different stress pathways, to understand the effect of the lunar surface radiation and the partial gravity of the moon and how that affects biology. The second one, we're going to talk about now bioproduction or biomanufacturing. We want to understand the effect of this lunar surface environment on the production of something, in this case, antioxidants or carotenoids. The third one is how the lunar environment, how the biology responds to the lunar environment using what is called genetic countermeasures. So we're bioengineering new yeast strains that allow us to make them more robust or stronger to desiccation or to the lunar environment. So we can guarantee that in the future, we can send these biotechnology type of yeast so they can produce something of interest for the astronauts for long-term missions. And the fourth one, of course, is to understand the space radiation environment on the lunar surface. So we have two different instruments. One is a very similar LET spectrometer to the one that we use on BioSentinel to detect charged particles on the surface. But also we are carrying now what is called a fast neutron detector. Now on the moon, when the galactic cosmic rays hit the moon that lunar regolith, those charged particles bounce back as neutrons. And neutrons, even though they don't carry a charge, they could be very dangerous to biology, and we don't really understand much of the effect on biology. So these two instruments will allow us to detect that type of particles and how this correlates to our biology experiments in AIMS 1, 2, and 3. Biomanufacturing, why do we care about this? So we are interested in sending astronauts for long periods of time to the moon or to deep space or to Mars. And we believe that we can use model organisms to produce bionutrients or to produce medicine or foods, not just using microbes, but also other organisms like plants or even algae that are known to be able to produce things of interest. This is related to what is called the bionutrients projects here, also at NASA Ames. They have been using a variety of organisms like yeast and algae to produce chemicals of interest like foods or carotenoids, antioxidants. So our main interest, as I mentioned over the last few slides, is to develop a capability. But we want to develop a capability that allows us to not just use it on the moon, but on Mars, but everywhere. Something that is highly flexible, that we can put it in a satellite, that we can put a lunar lander like Leia, maybe on Mars, on the ground on Earth. So it allows us to study not just one organism, but a variety of different systems. For example, we know that we can use this biosensor instrument to grow plants in fluids. We can also grow algae, cyanobacteria, and we're trying to figure out ways that we can even extend this to be able to do organoids or human cells. Some things need to change, but the idea is always the same, trying to use the same type of system so we don't have to keep changing again and again and again, that we can use it in many different types of planetary bodies or satellites or a lunar surface, etc., etc. Well, that was it. That was a quick presentation. I hope you enjoy learning more about model organisms and how we're using them, not just for biomanufacturing or simple studies, but also for deep space missions to the moon or interplanetary space. I leave you with this image of Artemis 1 launching, but also you can use that QR code to find where BioCenter is located and how it is right now compared to the moon or other satellites. Thank you very much, and I hope to talk to you guys soon. Bye.